All right. Hello, everyone. Good afternoon, depending on where you may be. Thank you for joining us today for the second of our ALS People with Lived Experience Forum of the year. Today, we're very happy to be joined by Dr. James Berry, Dr. Jinsey Andrews, and Dr. Bob Bowser. So we have three of the principal investigators of the Access for All in ALS Clinical Research Consortium. So thank you all for taking the time to be here today, Drs. Berry, Bowser, and Andrews. If you recall, back in February, Dr. Berry joined us to present sort of an overview of all ALS and how things were developing and evolving in those earlier stages. So fast forward to now, today's discussion will feature a bit of a deeper dive into the updated protocols for the study, as well as patients, recruitment, and outreach efforts. Before I hand it off to our PIs, I'd like to remind you that this webinar is being recorded. And also, if you have any questions, please type them into the Q&A box, and our panelists will do their best to get to them all at the culmination of the presentation. Any questions we are not able to get to, we'll do our best to follow up with and get to those with a follow-up email. So we'll devote the last 15 minutes of the hour to the question and answer session. Dr. Andrews, Dr. Bowser, Dr. Berry, the floor is yours.
Thanks so much, and thanks to everybody for being here. It's exciting to get a chance to talk about this project. It's something we've been putting a lot of time into, and many of you have also been a part of the design of this. And so we're very excited to move from the design phase to the operations phase. And I'll just start with the really exciting news that we have IRB approval for these protocols now. That just came through in the last week or so, and really clears the way for us to move forward. So really exciting time. Next slide. It's also, I mean, I think it's also an important time to just note that AMP ALS, all ALS, and I'll sort of explain the relationship of those, but all of these projects really harken back to support from the Access to Critical Therapies for ALS Act, or the Act for ALS. And this really grew from a groundswell of support from the community. I think it was, people with lived experience and academics and people really across the community that push for this, and it's doing what it's supposed to do, and that is moving the science forward. So all of AMP for ALS, all ALS, and many other initiatives are actually being supported by this. Really, really important to keep an eye on sort of this kind of support. Next slide. So the Accelerated Medicines Partnership Program, and thanks to Amy So for these slides, is actually a program that cuts across diseases. It's really an approach to sort of bring together communities who are invested in various diseases, bring funding, and bring research priorities, and then act on those research priorities, with the idea that we'll understand diseases better, but not just to understand, but really to move forward drug development. And to be able to add ALS to the list of diseases that are being supported by AMP ALS or AMP programs is really exciting. Next slide. So I'm using a sort of for many of you will be very familiar with these acronyms, some not so much. But there is a relationship between AMP ALS and all ALS. So AMP ALS is this sort of overarching broad view goals of trying to move forward ALS science and give the tools to the community that we need to develop drugs faster. And all ALS is a part of that initiative, but it's a very specific part. So all ALS is a consortium of sites, and you'll see the structure of this in just a minute. And it's really there to help implement some of the research priorities of the AMP ALS Steering Committee, and to provide critical scientific input and design features for sort of how we get to certain goals. All ALS is an infrastructure to enable large-scale collection of clinical data and biospecimens. That is, fluids, blood, spinal fluid, which are really valuable for us doing research in the community. And those are things that can be shared alongside clinical information and get us much closer to understanding and be able to target the key problems going wrong in cells in people with ALS. Now, in order to really support this, there is a centralized data knowledge portal that's being built. So this AMP ALS knowledge portal will ultimately receive all of the data from all ALS, and that will be shared broadly. And BioSend is an NIH biorepository that will receive all of the samples, and they will be overseen by a committee that shares them, and they'll be safe at the BioSend biorepository until then. One of the key things about the All ALS program is that we know that the AMP ALS steering committee may sort of identify a new priority, and All ALS is ready to sort of respond to those new needs. Next slide. So All ALS is a consortium. It's really created so that we can have a flexible research group that's working together to sort of be able to achieve these goals that may grow and modify over time. We want to provide opportunities for anybody living with ALS in the United States to participate. And you'll see some of the design features that we've put in place to accommodate that. We want to run longitudinal natural history and biomarker studies that are so important for us understanding this disease and having the baseline information we need to design better trials, to sort of implement better outcome measures. We want to build on the already existing large openly shared information, put that together into a portal and a biobank, and really have this be sort of a shining star for the community. And then, we want to ultimately provide clinical data and biosamples to researchers. Next slide. So the structure of this looks something like this. There are probably some nuances here, but AMP ALS and the steering committee and the priorities of AMP ALS are sort of at the top. Working as a part of that program is the All ALS Consortium. This is a group of sites that have agreed and contractually are working together, will implement protocols, can have more than one protocol, can accomplish more than one thing together. And I think really, in general, these sites are very excited to work together. Those sites, we need a structure for those sites. And so we've divided the sites into those that are sort of under the Barrow Neurologic Institute and those that are under the Healy Center at Mass General, reporting for sort of coordinating this study. And so that's where sort of the direction comes from. There are 19 sites under Barrow. There are 15 sites under Healy. It's actually now 16 because we're including the NIH as a site. So we're already growing, which is exciting. We have shared protocols across the consortium. So whichever center is sort of the coordinating center for a given site, we're working off of shared protocols. We have a shared safety monitoring board, observational study monitoring board, OSMB. We have shared steering committee, and then we have site monitoring that happens through BNI, Barrow Neurologic Institute. Now, site monitoring is essentially sending an independent reviewer out to each site to look at all the data, frankly, to make sure that we're not making errors in how we collect the data, and to make sure that we're following all the rules. And I think it's actually really important. It is very easy to have transcription errors, to miss an important data point. And this happens all the time, and the monitors are a big part of making sure that we get that data. All of the data will be collected through a shared data coordinating center, and we're using a single IRB, which simplifies the process a lot. And ultimately, we'll have the data portal and biorepository, which will be overseen by the AMP ALS group to make sure that data and that the samples get out as broadly as possible. Next slide. So Bob Bowser and I are what are called the contact PIs. So we're sort of the ones that are interface with NIH the most. But we represent a large group of multiple PIs. So this is kind of a group of PIs working together. But I think one of the largest groups that I've worked in, because it's a very big project and we wanted to have lots of expertise. Next slide. So these are the other MPIs, really, I think, huge expertise across the field, close colleagues, and really excited to set direction for this project. And it's a sweeping scope, this project. So it's not so dissimilar it's not sort of like a brand new thing to do to collect clinical information and biosamples, but the scope of this really changes it. And there's a lot of work that we've been doing to make sure that that works right. Next slide. So the coordination center teams at what we call the East Coordination Center, MGH, and the West Coordination Center, Barrow Neurologic Institute, have been incredible. I think, Bob and I are very much aligned in saying that the progress that we've made would never have happened without these teams. They're growing. We may actually have a few more names to get on here. But I mean, just remarkable teams doing incredible work. A lot of experience, a lot of energy, very responsive, just really a joy to work with. Next slide. So I'm going to hand over to Jincy Andrews to talk a little bit about the sites and the recruitment efforts that we put together.
James. So I think the next portion is really to talk about our real thoughtful process of trying to make sure we cover a large geographic area in this consortium that we're trying to make available to as many people living with ALS as possible. And so as you can see on this map, we've tried to cover really the entire United States to have a center in each region and really have both the West Coordinating Center coordinate some of the centers and the East Coordinating Center to coordinate some of the centers. So there's a total of 34 clinical sites. And if you go to the next slide, we kind of show, and if you click through, we have many experienced clinical researchers that are leading the efforts at their centers that have shown a lot of enthusiasm about working on this project and really willing to go the extra mile to ensure that we can meet the project goals. And again, a lot of these centers have participated in many observational and clinical trials and also cover a large swath of the United States so that we, we've been very thoughtful about trying to make this as accessible as possible to as many people living with the disease, because this is, as James said, a very large project. And the next slide reflects the ambitious goal. If you click one more time, the ALS enrollment objective is really to enroll more than 2,000 participants, and we have ambitious timelines of including these participants as quickly as possible. And so it is one of the largest coordinated consortiums for this kind of activity. So it requires really study design and methodologies that we really haven't used before in ALS research. So that's part of the innovation of all ALS in this consortium. But when you innovate, it takes a lot of work and building it. And so that's what the team and the multiple PIs have been working with, the contact PIs, to try to make sure that we think through each step. And so if you go to the next slide, one of the things that we really want to make sure that we think about as concepts and themes while we try to enroll these participants is really to think really large and be as inclusive as possible. So we really want to make sure that we get a diverse population. This is one of the areas that I'm trying to focus on as well as part of the team, in trying to make sure we get people that are maybe in underserved geographic areas, include a diverse race and ethnicity background of participants into the study, and try to capture a variety of socioeconomic status and education levels to get a real representative population for ALS. But in doing so and trying to do this, we also recognize that there are barriers to participation that we've talked about in our community, and we've heard from a lot of people living with the disease and also from their caregivers who have to bring them. So we really try to learn from the community and hear about and get informed about ways to kind of reduce the barriers to participation. And then, of course, in our community, for many years, we've been talking about smaller scale studies and not wanting to duplicate and reinvent the wheel. So one of our real goals and objectives of this consortium is to coordinate with other ongoing studies like Target ALS, the Natural History Study, and the Prevent ALS Study. And of course, one of the main things that will drive the success of all ALS and meeting our goals and objectives is really making sure that we work very heavily with the community and organizations that interface with people living with the disease, and make sure that we are really attentive and really coordinated with our outreach activities and communications. So community engagement is a real key portion and a critical portion, which I believe would help us meet our objective of enrolling the number of participants we need for this consortium. If you go to the next slide, one of the really neat things about all ALS is that we're using on-site at clinic visits, but we've also tried to really think thoughtfully about decentralizing and using study methodology that actually allows people to perform some or all of these activities remotely. So we have, for communication purposes, recruitment materials, and we'll use our nonprofit partners in the space to help communicate the study and ways that people can engage and try to participate. We have a dedicated All ALS website, but some of the decentralized methods include using an informed consent that can be done remotely. So we call that the e-consent, instead of having to go to the center and interface with research staff in person at a clinic. We're also incorporating patient-reported outcomes being collected using NeuroPro, and also using video conferencing and using apps that can be used in the home to collect some of the assessments. And then really one of the challenging things is really trying to make sure that we can get samples from individuals that are participating and trying to get home phlebotomy services and collections done remotely to enable this kind of decentralized remote participation portion of the study. James, did I miss any of the methods there? Did I cover all of them? And so, yeah. All right, thanks. So then if you go to the next slide, I think this is one of the things I love about this consortium is that the All ALS consortium really has a lot of advantages. I think this is like the first time in a very large scale that multiple investigators and people living with the disease have worked very together, and other stakeholders, to really think about what are the barriers to participation in research and trying to eliminate those. We're also trying to do large and expanding numbers of participants to enable rapid capture of data and biosamples. So we're using a lot of different types of measures, and maybe not everybody does the same measures. So there's speech measures and digital measures for limb movements, for example. And there's a lot of flexibility. So James highlighted earlier in the presentation that we're building this consortium, and part of the importance of building this consortium is not just the large scale, the decentralized methods, and collaborating with ongoing initiatives and trying to synergize in the field and catalyze these things, but also to be flexible so that if we employ these and changes need to be made, or we need to add more sites and add more participants, we can do so. And so we have the ability to quickly add more research protocols when requested by AMP ALS or the research community. So that flexibility is something that's really important, and I think very novel for this type of a. If you go to the next slide, I think I'll just continue on here. If you go flip one more, I'll take the recruitment and outreach level. So communication, as I mentioned, at the site level is very important. So we will work specifically with all of the investigators at each of the sites to really utilize their local networks to interface with people living with the disease. So what we want to do is really get to patients where they are. And like I said before, we are going to kind of partner with nonprofit partners and advocacy groups to communicate about the study, about how to enroll or engage with the study, and maybe even discuss milestones. So this really is a collaborative effort to try to make sure that we connect to as many people. And it is a work in progress. It may be that the things that we employ now may reach some people, but not all people. And so part of the process in this consortium is not only utilizing the tools that we're aware of that may work, but also thinking about and communicating about ways that we might implement new techniques to communicate to people that we may not have reached initially. And some of those tools actually on the next slide for the consortium level include materials really dedicated to help support recruitment. So there's a dedicated All ALS communications and outreach coordinator to build relationships and raise the consortium's presence. So having dedicated personnel for a particular project, especially in ALS, has been really a key driver for success in any kind of research project or clinical trial. Also, a dedicated All ALS patient navigator will help direct remote participants to individual sites. And this this is something that we've heard from the community over and over again about having someone help them so that they don't have the burden of trying to find out how to do it, who to contact, which site to contact. So having that All ALS Patient Navigator will facilitate some of that movement of potential participants to sites and projects. All ALS website will also be used to engage potential participants and drive the awareness. So that website is something that I think will be key to help people stay informed about the study and give a tool where people can access it and figure out a way to engage with the All ALS Consortium, especially locally at their site. So if you go to the next slide, while we're trying to engage the community, communicate about the study, I think this is a moment due to the scale of All ALS. We really have an opportunity to focus on equitable, diverse, representative enrollment. We really want to make sure that we do this right, and we do it in a way that really represents the entire population of people living with ALS. And so we know that most of our clinical trials have not done a really great job. And it's not that they weren't trying to do a good job, it's just there are, like I said, barriers for participation in research and barriers to access that's variable across the country. And so one of the things that we really want to do is set goals for sites to really think about who's participating and really try to account for diversity of enrollment of participants. And we also want to make sure that when we're launching this and moving into the active phase of All ALS enrollment, we want to make sure we're communicating in a way that's appropriate for the regions and the populations across the country, support those communities, involve liaisons that include that, and build from sites, especially the sites in those particular regions, will be very familiar with their patient population. And so we really want to make sure that we focus on that. And, you know, as we try to focus on these types of maneuvers to try to make sure that we have an equitable and diverse representation of participants, there may be opportunities along the way as we look into means by which we can evaluate the population that's enrolled and try to look at different variables and include people who specialize in diversity and equity inclusion in clinical trials. We'll likely implement more tools to kind of quantify where our enrollment is coming from, how the population looks, and we may implement changes going forward. So that's part of a really focused goal in this All ALS Consortium. If you go to the next slide, you know, these are some of the ways that we can operationalize equitable enrollment. So I gave some examples, but we really want to talk about actually how to, you know, where are the gaps in equitable enrollment. So we talked about participation barriers. For example, we want to really accept the fact that we could do a better job and reflect on potential ideas and maybe even get input from people that have worked heavily in this space and have implemented changes and factors that we can quantify and improve the enrollment. We want to engage with groups focusing on diversity and equity. We want to build the community infrastructure, and we really want to stress this as a particular goal to sites. So, you know, usually in clinical trials and research, we stress milestones like the number of participants enrolled, but implementing a goal of being diverse in their enrollment and recruitment will be an objective for individual sites. And of course, we want to make sure that we dedicate, and I'm part of that team, to dedicate a focus specific to operationalizing this. And, you know, so that includes identifying the gaps, trying to implement ways that we can improve it, and then actually evaluating as we hit milestones in enrollment within the consortium and really report out any kind of progress as we enroll participants into this consortium. And if you go to the next slide, while we're trying to diversify the participants that enroll in All ALS, like I mentioned before, we really want to make sure that we're not duplicating or working in parallel to existing efforts, and really want to make sure that we're communicating and collaborating, which are key concepts, I think, that make All ALS very special. We want to make sure that we're working with efforts. For example, Target ALS, we've already incorporated some of the efforts that Target ALS has been doing by incorporating those sites into the All ALS Consortium. We've already integrated the Natural History Consortium, and we have funding from the same source as All ALS, and we've also partially integrated the CDC registry, and we're working on that to kind of harmonize data collection and linking all of that data to a global unique identifier called a GUID. So I hope that I was able to convey one of the unique aspects of All ALS in terms of trying to use innovative methodologies to be inclusive by including decentralized methodologies. I hope I was able to convey that we're really dedicating a team focusing on equitable and diverse enrollment and participation in All ALS to reflect a general population, and that we're really trying to collaborate with existing efforts and synergize those efforts so that we're not recreating the wheel. So with that, I'll turn it over to James to talk about specifically the study protocols. As he mentioned earlier today, we did get IRB approval, so I'll turn it over to James to talk about the specific protocols.
Thank you so much. I mean, that was fabulous. I think this, so, I think the parts of this program that Jinsey has just talked about, which are, really building community, building sort of shared vision, are going to be really important as we move through this project, because that's, I think, how we convey to everybody that this is really meant to be something for the entire community, and collaboration is going to be key. And I mean, just a huge thank you to all of the groups that have already, all of the groups and all of the people who have already provided us with ideas, given us great insights about how to set up a project like this, and just sort of jumped on board and said, we'd love to be helpful in getting this done. So we are excited to continue that, and that'll become a real focus now that we've gotten through some of the initial approvals for the protocol. So the All ALS Consortium is just that. It's a consortium. It's a network of sites that are sort of bound by this idea that we will do these studies together, and that we want to advance science, and that we want to do this using principles of what we call open science. And that means essentially the data that we collect, the samples that we collect, are going to be shared. And, it's not that Bob or Jinsey or I expect that we'll have any special access to this. So we really want to do this for the community, and then, to use a word that's sometimes overused, I guess, crowdsource it, and allow as many advances to come out of this as possible. In order for us to actually do anything as a consortium, we need to have study protocols. And so I'm going to talk about two study protocols. One is called Assess All ALS. And this is for people who have symptomatic ALS, as well as people who will act as controls. That is, they don't have ALS, they don't, they're not developing ALS now, they don't have a gene that may cause them to be at high risk of developing ALS. So that's Assess All ALS. And that's a study that we've actually done, it's built on methodologies that we've used many times over the years in smaller studies. And scale makes a big difference here. And I think our decentralized approaches will make a big difference. But this is maybe some of the methodologies well trodden. The second protocol that we put in place is called Prevent All ALS. And this is built on really projects that are happening at single centers at a few places in the country, maybe two centers together. And this is sort of looking at people who are or may be carriers of gene mutations that can cause ALS, and really trying to understand what that pre-symptomatic space is like, what's happening biologically, what's happening with symptoms. And this is a little bit different. Now, as I said, there are a number of centers that have done this as a single center or as one or two centers, but to roll this out then to 35 centers is a very different thing. And it's taken a lot of work, and a lot of work from people with lived experience, a lot of work with people on the team. Michael Benatar is not formally a part of this effort, but we owe him a great debt of gratitude. He has been doing this for many years, and has really led the way in thinking about this. I think it's important that we mention that. So I think we'll learn incredibly important information from both of these. Next slide. I'm going to talk first about assess all ALS and sort of give you the study overview. So the primary objectives, so when we think about, what are we trying to do? We call it objectives. Some people use goals, but our primary objectives here are to establish the necessary infrastructure, policies, and procedures to recruit a significant portion, and that's the language that we've been using, of people with ALS in the US into the all ALS study. This really means a lot of the legwork we've been doing over the last 9 months is to create the mechanism to do this. We want to enroll participants into all ALS using in-person and remote strategies, and then really ensure that we are hewing to the principles of diversity, equity, and inclusion. Those are really important to us. I will just say candidly, we have not done a good job of this in previous ALS studies, and I think we need to pay real attention to it. There also may not be very easy answers for this, and so we need a lot of new approaches to really do a good job of this. We're going to follow participants for 2 years, and we're going to measure a wide range of ALS outcome measures. The bedrock of these outcome measures are ones that we've done for many years, but we want to hang around those other sort of outcome measures, other things that we can measure that tell us about additional portions of the disease. And then in association with all this data that we're getting, we want to collect this robust set of longitudinal biofluid markers. And this was, I think, a question that came up in the Q&A. We'll be collecting these over time. We're going to focus on primarily blood and spinal fluid, and because those are some of the rich sources for biomarkers. Now, blood can be processed many different ways. We can get whole blood, where we can look at the genome. We can look at plasma, where we can look at certain biomarkers. We can look at serum, where we can look at other biomarkers. We can get just the white blood cells, which can be used to create stem cells. So there are lots of biofluids, but they really, originate as CSF and blood. Next slide. So we also kind of want to explore what we call minimally important differences of the ALSFRS-R and speech. And what this means is we sort of, I have some slides where I talk about it, but essentially we're saying, what's the smallest difference that matters? If people say, well, I'm about the same, how much wobble can there be in the ALSFRS-R, for example? Or if they begin to say, well, I'm a little bit different, how big does that difference have to be? Because that matters when we're designing clinical trials. We don't want to find a drug that makes such a small difference that it doesn't matter to anybody. But at the same time, we don't want to throw away a drug that makes a difference that maybe not a home run in and of itself, but could be part of a drug cocktail and is having, enough of an effect that people would notice. We want to look at things like ALSFRS-R association and survival. And we hope that there will be many, many, many other questions that come from other people that they're investigating. And then lastly, we want to capture speech recordings and identify features that are different between ALS and controls and reflect ALS progression. And there's some really important legwork that has happened from a variety of investigators over the last 10 years to put us in a great position to have a big speech bank and move forward. Next slide. So the study design for assess all ALS, this is, again, we're in the symptomatic space here. There will be, if we look at the participant cohort, there are going to be people who have symptomatic ALS who decide to participate entirely off-site. Sometimes we call this remote. Of course, it depends on what your position is. If you're at home, you're not, what are you remote to? But so we think of that as off-site. Some will participate on-site at clinics, and then some will be controls. And I'll show you sort of at the end what the controls, how the control schedule is a little bit different. But either way, whether people are coming in for people who are symptomatic with ALS, whether they're coming in on-site or off-site, every 4 months we'll have a kind of a visit, whether it's a video visit or an in-person visit with them. And there's a big window on this. We know that it won't happen exactly at 4 months. It could be a month or more in either direction, and that's okay. We're going to collect clinical outcomes, and then we're going to collect something called electronic patient-reported outcome measures and speech. And I'll show you, those are digital measures, and I'll sort of show you what I'm talking about in just a moment. And we'll collect biosamples, even from people who are participating off-site. We're going to have a mechanism for home phlebotomy, as Jinsey talked about earlier. All the data goes into a central electronic data capture system or central database, and then it will be sent over to the AMP ALS data portal, where it can be can be shared. The biosamples will be at BioSend, and they can be shared from BioSend in conjunction with this rich clinical data. Next slide. So again, we mentioned the 2-year follow-up. We mentioned that all participants will be seen every 4 months. They will all have what we call a clinician-administered ALSFRS-R. This is kind of our traditional way of having somebody ask questions and sort of get to the ALSFRS-R answer. There will be a blood draw that'll either be done on-site by a coordinator or a nurse, or it'll be done remotely using home phlebotomy, depending on how people are participating. And then all participants will also have a cognitive assessment called the ECAS, which has been used in a number of ALS studies. It assesses especially frontal and temporal dysfunction. And we'll do that at baseline 12 months and 24 months, which is about the frequency that it's been designed to be delivered at. This covers things like what we call executive function, which is kind of organizing thoughts. It does cover memory, language, which we think is important, visuospatial skills, and social cognition. And it is, we've talked a lot with our friends and colleagues in the frontotemporal dementia space. We know that there is some overlap between these. It is not the assessment that they use, but we are working on ways of sort of harmonizing the data that comes from this with the data in the FTD world. It is being used in large, similar efforts in Canada and in Europe. And so, you know, we do gain kind of some harmonization with those efforts, which I think is really important. Next slide. For those who are coming in person, we'll also get vital capacity. Handheld dynamometry is a quantitative measure of strength called HHD. We're going to do that in 3 muscles in the hands. Those are the ones that seem to change most rapidly. And it's this balance between what can be done efficiently with lots and lots of people versus being, you know, completely extensive. It might limit the number of people that we can enroll. And then cerebrospinal fluid, or CSF, will be collected from a subset. This will be optional. And we are really working closely, and not only just working closely with, not only just collaborating, but really integrated with the Target ALS study, which is going on at many of the centers that are participating in all ALS. And that is a very close coordination. Barrow Neurologic Institute and Bob Bowser are the leads of the Target project, and it was really part of the proposal to do these hand in hand so that the data, the samples will be harmonized and can be shared at the same time. Next slide. So I mentioned the digital assessments. Again, this is for the symptomatic group. So these are electronic patient-reported outcomes. They will be collected on a system called NeuroPro. NeuroPro is a web-based interface. There will be a reminder that goes out by email. Everybody will have sort of a window that will be open for say the first 10 days of the month. And during that period, the centers will know to remind people. The reminders will go out from NeuroPro, and NeuroPro will be sort of open to accept the answers to these. We have a schedule so that we're certainly not giving all of these all the time, but we are sort of interdigitating so that there'll be 1 or 2 surveys that people answer about once a month. We're trying to limit the number. There will be a few more at baseline, so we know it'll take a little bit longer. And we're trying to give people a sense of how, you know, what they're getting into and allow them to do it in chunks and come back and finish if they need to. Next slide. There'll also be a speech assessment that will be done where people go through a battery of recordings. We're doing this in conjunction with Oral Analytics. We've worked with them on both observational studies as well as clinical trials like the Healy platform trial in the past. It's gone very well. Every 3 months, we'll do what we call a full battery, which is sort of more tasks, more recordings. But the motor speech modules will take about 4 minutes, and that'll be done once a month. Next slide. For controls, there are going to be 3 visits. A baseline visit where we do many of the same things that I just talked about, basically all the same things that we just talked about, except ALSFRS-R. And then annual visits where we do a sort of a restricted follow-up so that we have some longitudinal data to compare, but we're not sort of putting the same amount of time into controls and people with ALS. There's an opportunity cost. In other words, we want to capture lots of people with ALS, and we want to have some controls to compare to. Next slide. I said I would talk briefly about minimally important difference in outcome measures. I'm going to go through this very quickly so we can get to the prevent ALS. But basically, we're asking how much change is meaningful. Next slide. So we do the ALSFRS-R at baseline, and then 4 to 6 weeks later, we at the second visit say, have you had? It has to be close enough that people can kind of remember. Say, have you had any important change? And then if people say yes, I have, then we have them rate on a very simple scale, a little bit worse, a tiny bit worse, somewhat worse. And that gives us sort of a ground truth. And then we can compare the difference in ALSFRS-R to this sort of overall difference and begin to see what we find. Next slide. So Christina Fournier, who's at Emory and a very creative researcher, I think has done this both for the ALSFRS-R and Rhodes, a smaller group of people. And what we'd really like to do is use this All ALS forum to really validate, not duplicate. It's not wasteful. It's actually a very thoughtful validation of her original work. And we'd be happy to work with her on this analysis as well. So, she has said that about an 8% decline in the ALSFRS-R correlates with a little bit worse. And if we can validate that, I think that's a really meaningful thing for us as we design trials. Next slide. Next slide. There's just a little more detail here. So I want to have a few minutes to talk about the Prevent All ALS study. The primary objective here is to characterize people who are asymptomatic, don't have ALS or FTD, by obtaining natural history prior to the onset of symptoms. Some of these people will never develop symptoms, and by performing longitudinal follow-up in these people who are genetically at high risk of developing ALS or FTD. Now, not everybody is as familiar with this, so I'll just say that we have a list of mutations that are known to be causative of ALS. Interestingly, not everybody who has those mutations develops the disease, so it's not what we call 100% penetrant. And that's a bit of a challenge for us, but also an opportunity. If we find people who are older and have not developed ALS, we can ask ourselves why, and that's a very, very focused question. So really, really important information here. Some people with some of those mutations are actually at a high risk of developing frontotemporal dementia or ALS or both. So we are going to need to work very closely with the frontotemporal dementia world, our colleagues, in this study where some people may develop symptoms of FTD or ALS. Our secondary sort of objectives here are to create this cohort of genetically at-risk individuals. This can be really valuable. Let's say, for example, there are trials that begin that are prevention trials. This would be an incredible way to sort of let people know about those studies so that they can take advantage of those opportunities if they'd like to. We can also, in the context of this study, offer ALS gene testing with appropriate genetic counseling and do that outside of the clinical record, which is sometimes important for a variety of reasons. We also then are able to follow people and really evaluate them rapidly if they do develop any symptoms, which is something we sometimes call phenoconversion. Next slide. So again, I mean, this is a prospective observational cohort study. We're not following people for 2 years. We're really just following people in this one. And we'll follow them as long as they're willing to be a part of the study. We'll get clinical outcomes. We'll do a little more detailed cognitive testing in this study than in the ASSESS study. We'll do the ePros, similar to what we had just talked about, but less frequently, and then collect CSF and blood. Next slide. So here, a person who might be genetically at risk for developing ALS will come into the study. The protocol is hybrid. There's an annual on-site meeting. There are also at-home visits to get home phlebotomy a little more frequently. And as I said, there's an ePro component to this as well. And then data will go to the central EDC. It'll be shared through the knowledge portal, and the biosamples will be shared at BioSend. Next slide. So the way this looks is really sort of at the screening visit, which is visit 1, it'll be done in the clinic. And then there'll be visits quarterly, every 120 days, remotely at home with home phlebotomy. And then people will come in every year. Now, if somebody wants to leave the study, they can always leave if they're willing to come in for what we call an early termination visit. That is, they've decided to leave the study early, then we would have them in as possible. They don't have to do that visit. If somebody thought they were developing symptoms, we could have them in and do kind of what we call an ad hoc visit to assess them. Next slide. I'm not going to go through all of this, but we really detailed clinical information, more detailed cognitive assessments of the ECAS, but also a more in-depth evaluation called FTLD-CDR. We'll draw blood, and then there is a sub-study that I'll talk about just in a moment that allows genetic testing and return of results to the participant. Next slide. So the remote follow-up visits will happen at home. There will be PROs. There'll be a symptom questionnaire and a blood draw, and we'll be available to talk to participants anytime. Next slide. Similar assessments in person to assess. Our handheld dynamometry, for example, will be every muscle because we really need to have a good characterization of strength in people, but otherwise quite similar. Next slide. So there is going to be a genetic testing results sub-study. So this is essentially a way that people can come into the study. If someone is from a family where ALS is common, then they could enroll in this study even if they don't know their genetic status. So they may or may not be at genetic high risk for the disease, but they can come in and get genetic testing through this study. And that's done in a research setting, not in their clinical chart. It is confidential testing. And the components of this are really a remote screening visit with a genetic counselor for pre-test genetic counseling, genetic testing, which actually we've moved away from blood and moved toward a saliva collection. So a kit will be sent to the person's home, and they send saliva in. Those results will then be returned at a visit with the genetic counselor, and then there'll be a follow-up check-in visit. Next slide. So I think this just lays out what we already sort of went through and a bit of a timeline for when those visits will happen. Next slide. So we have a particular focus on what we call phenoconversion. That is, we want to support people who may be developing symptoms of FTD or ALS or both. And so we have sort of a mechanism to see people at the site near their, where they're participating, hopefully near their home. Anytime they think that they're developing symptoms, obviously they can get in touch if there's a question. They can be seen for a visit. We have outlined some fairly basic things that should happen from a research perspective, but this is also a very highly emotional, challenging clinical situation. And so there may be many things that happen to clinically take care of a person in this situation that will happen not directly as a part of the study. And that's really the priority. We want to make sure that we're taking care of a person. This is also a time where we can get rich clinical information for research, but all of that is subservient to taking care of the person, making sure that the person is cared for. Next slide. So just briefly, we're out of time, so I won't go through all of these. We've made some amazing initial steps at design. We've set up the coordination center and the data coordination center and all of the sites. Contracts are in place. We're contracting with vendors, so there's a lot of vendors we have to pull together, doing home phlebotomy, doing speech assessments, some of the kind of consenting processes that we've put in place will require vendors, and so on. We've put together governance about publishing, about sort of our standard operating procedures, about sort of oversight of the study. Those are fundamental. We've really spent a lot of time doing outreach, and we know we have a lot more outreach to do. Protocol development went very smoothly. There was a lot to work through. Many of you on the call may have been a part of that. The IRB has now approved the protocols, as we said. The electronic data capture system or just study database is being built. We've sort of put together what that will look like, and it's sort of being built and validated. And we're really hoping to continue to build community around this study and the ones that are collaborative with it. CDC, the natural history study, existing data like Answer ALS and GTAC. So really, really exciting. Next slide. This is something that we talk about a lot, that, we all want this to culminate in treatments and cures for ALS, and we are confident that that will happen. How we climb this mountain together also matters. We need to build a community of trust. We need to do our best to do this right. We want everybody to feel comfortable and confident that we are putting together a study that is meaningful, and we want to feel that way ourselves. And that's the climb, and that climb with a team and a community like ours, I think is really something else. Thank you.
I think we are right at the hour. Dr. Berry, Dr. Andrews, Dr. Bowser, any last words before we wrap up?
I just want to thank everybody for being here. It's really a pleasure to work together, and for your offers of help. So Katrina, I see your offer of help, and I have your information. But thank you to so many of you for being here. I think spreading the word, learning more about this so that you can, you can give feedback, or you can, hopefully advocate for the project, and just thank you for participating.
All right, I think we all echo that. Thanks again to our panelists, Dr. James Berry, Dr. Jinsey Andrews, and Dr. Bob Bowser for taking the time. And thank you everyone who joined us as well. We will reconvene the forum again in a few months, and we'll hope to see you then. Thanks everyone, and take care.